While the Calvin cycle is probably the most famous carbon fixation pathway, there are others that we need to at least mention, although I don't expect you to know a whole lot about these in detail, primarily just that the Calvin cycle is not the only variety of carbon fixation. So another reductive cycle is the reductive or reverse TCA cycle. And so what this does is run the citric acid cycle, or I guess I could have called that the TCA cycle to make it a little bit more consistent, basically in reverse, allowing you to reduce carbon dioxide to acetyl-CoA. So remember, typically when we talk about the Krebs cycle, we are fully oxidizing all of the carbons that are originating from like glucose, and they're basically converted into pyruvate, which is converted into acetyl-CoA and kind of pushed into the Krebs cycle. And this is the point where all of the carbons from glucose get fully oxidized and exit the cell as carbon dioxide. So this is exactly the inverse of that. And it's very common in chemolithoautotrophs, and it's the second, as far as we know, the second most common pathway to see in the microbial world. And so basically what this is doing is using the same enzymes as the TCA cycle. You can see here the so-called normal TCA cycle here, and here is the reverse TCA cycle, except you're expending a couple of molecules of ATP. So just like in the oxidative TCA cycle, how you generate some ATP, here you're going to have to apply some ATP to fix carbon dioxide. So very similar compounds, very similar enzymes. One other thing to mention here is when you're doing the reverse TCA cycle, it provides several other intermediates. So depending on the needs of the cell, you might not need acetyl-CoA, maybe you need some other things. And by funneling your carbon dioxide through this pathway, you can generate those other things and kind of stop the cycle wherever you need to, if that's what's necessary for the cell. Alright, so another option is the reductive acetyl-CoA pathway, and this is used by methanogens. Remember, that's only found in archaea. Only archaea are capable of generating methane, as well as anaerobic bacteria such as the sulfate reducers that are conducting anaerobic respiration. This basically involves several unusual enzymes to follow portions of the acetyl-CoA pathway for carbon fixation. So this one's interesting because it's not necessarily like blending an existing pathway and capable of going in reverse direction, but instead using completely different enzymes to produce this intermediate. So this kind of goes counterintuitive to what we think about being like efficient in terms of the genome. You'll have to use these brand new enzymes that are only used for this. Still, it's one of the pathways that we see in the microbial world. Some other ones we see, we have the 3-hydroxypropionate 4-hydroxybutyrate, that's a horrible name, which has only been known about for a little over a decade. And this is commonly used in thermophiles, bacteria, and archaea. Probably some form of horizontal gene transfer happening, since we only really see it based on locations. And so again, this has a very unique reaction sequence to produce a compound that's used as an intermediate or a precursor for other forms of biosynthesis. Alright, and our last one, the hydroxypropionate bicycle. This is used by some archaea and some green non-sulfur bacteria, and these are ones that are going to be doing anoxygenic photosynthesis. Basically, you're producing another compound that's a precursor, that 3-hydroxypropionate, from carbon dioxide. And so the things to notice with all of these is that you have this common theme of actually reducing carbon dioxide, which is energetically costly. When possible, it seems that the majority of organisms will use enzymes that are anaplerotic, so they can go either for catabolism or anabolism, but some other organisms have evolved these completely unique pathways that aren't found in other or completely unique enzymes that aren't found in other pathways. So while that form makes much more sense from an evolutionary standpoint, this is what we observe, is there is actually a variety in the world of these different pathways for carbon fixation.